Okay, so we're going to pick up our discussion here today with diffusion. We began by looking at the structure of capillaries last time, and today we're going to move on to look at exactly how movement of materials occurs across the capillary wall. Okay, that's going to include looking at Starling's forces. Now, when we spoke about bulk flow so far in the course, we were looking at bulk flow in the context of our overall circulation, right? Our overall physiology. But we can also look at bulk flow in the context of movement of fluid across the capillary wall, okay? And so the flow of fluid from the capillary over into the interstitial space and vice versa is governed by that same pressure gradient that we spoke about. And so we have the movement of protein-free plasma. Remember that the capillary is impermeable to large molecules like cells and proteins. And so only fluid is moving across the capillary wall. And that gives us two forms of fluid movement. There's filtration, which is the net movement of fluid from the capillary out into the interstitial space. Whereas absorption is going to be the net movement of fluid from the interstitial space back into the capillary. So to really look at what direction fluid is moving in, we're going to have to look at those two net movements, but also look at the relevant forces that are acting on the capillary wall in total, okay? Before we get into the details, let's talk about why this happened. So we talked about last semester our two extracellular fluid compartments, the interstitial space and the plasma. And the movement of fluid at this level happens in order to regulate or to balance those two fluid compartments out. Okay, think about it. If we had fluid moving perpetually from the capillary out into the interstitial space, we would always be swollen, right? All of that fluid would pool in our tissues and we would have edema or just be swollen all the time. On the other hand, if we had fluid moving in the opposite direction, unregulated, we would have too much fluid in our plasma, in our capillaries, and that would not be good either. So the movement of fluid via filtration and absorption is necessary in order to balance both these extracellular fluid compartments out. Okay, so that's the kind of the why of why all of this happens and why movement happens. So let's look at what substances can move and the various mechanisms of movement. So we talked about the fat-soluble substances or the lipophilic substances like carbon dioxide, oxygen, some of our steroid hormones like thyroid hormone and so on. They can move through the cell membrane easily, right? They can diffuse. They're lipid soluble, and so they can get through that phospholipid bilayer easily. We can also see water-soluble substances moving through like our ions, sodium and potassium, glucose, although glucose is going to need the energy of another molecule. We talked about facilitated diffusion last time. So this is another mechanism of transport. But then we can also see transcytosis of some exchangeable proteins. And when we talk about exchangeable proteins, we're talking about things like lipoproteins that help to make our fats soluble, right? They move our fats around in the body, like that when you go to the doctor and they measure your HDL, LDL, that's what they're measuring, those lipoproteins. So these are some of the things that can go through the membrane, but remember, cells cannot for the most part, so red blood cells cannot, and proteins cannot for the most part. And in the context of the discussion, we will be looking at albumin is kind of the #1 protein that we're speaking about, and that is a protein that for sure cannot cross the capillary wall. Okay, we're gonna move on to speak about the Starling forces that govern capillary movement. Now, before we get into it, I'll make a note here. This has nothing to do with the Starling's law of the heart. They just happen to have been discovered by the same physiologist. But the Starling forces govern both filtration and absorption across the capillary wall, and there are two forces under these Starling's forces. The first one is hydrostatic pressure. And this is basically dictated by the fluid that's present in that fluid compartment. So if we're speaking about the hydrostatic pressure of the capillary, it's created by the plasma in the capillary. If we're speaking about the hydrostatic fluid of the interstitial space, it is created by the interstitial fluid. So the fluid that's present is creating that hydrostatic pressure. This is usually referred to as a push, and you're going to continue to hear me refer to it as that. And it's basically the ability to push or to force or to thrust fluid across the capillary wall. On the other hand, the second pressure is osmotic pressure, and this is created by non-permeating solutes. This is what we call a pull force. And when we say non-permeating solutes, we talked about this last semester, right? The osmolarity pull, and it's the ability of solutes that cannot cross the membrane to pull water to them. Okay, think about it. So you have a container, it's separated by a membrane, and your solutes that are dissolved cannot actually go across that membrane. So in order to get to diffusional equilibrium, those solutes will pull water to the side that has the higher solute concentration or the lower water concentration. We talked about that last semester. So a very similar phenomenon is what creates the osmotic pressure. And you'll hear me refer to this as either oncotic pressure or oncotic osmotic pressure, even colloid pressure, and those are all referring to the exact same thing. That's the pull of water to the side that has those non-permeating solutes, okay? Yeah. Okay, so let's kind of describe those Starling forces here a little bit better. Okay. So we're speaking about Starling's forces, and let's just draw a capillary here. And we're also going to draw a tissue at this top, right? So this is a tissue in question, and this is the capillary in question. And so we can label this as the interstitial space. Okay. And in the corner here, I'm going to put a key that describes our relevant forces. So P is going to represent the hydrostatic, and that is essentially a push. And pi is the symbol used to represent the osmotic, and we describe that as being a pull. Okay. Now, in the capillary, we have plasma. And because this is a fluid present inside the capillary, this exerts the capillary hydrostatic pressure. So that's our first hydrostatic pressure, the capillary hydrostatic pressure, which is represented by this large P and then CAP representing where the pressure is found. And this is a push force. So the direction of this force is out of the capillary. Okay, it's a pushing force, pushing fluid out of the capillary. Okay, and it's created by the presence of plasma in here. Now, out in the interstitial space, we have the presence of interstitial fluid. Oops. And this interstitial fluid also has a hydrostatic pressure. So we're going to represent this by, again, a large P, but then IF indicating where this pressure is found. Okay? And so the interstitial fluid hydrostatic pressure also exerts a push, but because it's in the interstitial space, it's going to be into the capillary, so this direction. Okay? So the capillary hydrostatic pressure pushes fluid from the capillary into the interstitial space. The interstitial fluid hydrostatic pressure pushes fluid back from the interstitial space into the capillary. And so these are opposing forces, okay? So that's our first set of Starling's forces. The other two forces, which are the osmotic forces, are pull forces. They're also acting on the capillary. So things are gonna kind of get a little bit complicated at this point. So we have proteins that are present in the capillary, as well as red blood cells, and they cannot cross the capillary wall. So they are exerting a pull force on the capillary wall. And so the first force we have is the capillary osmotic pressure. Okay, the capillary osmotic pressure. And that is a pull force that's moving from the interstitial space into the capillary. Okay, the capillary osmotic pressure pulls fluid from the interstitial space back into the capillary. All right, now we don't have many proteins out in the interstitial space, but we do have to take into account that force, that that interstitial space osmotic pressure. Lots of words here. Okay. So there's the interstitial fluid osmotic pressure, and that is also creating a pulling force. And so it's going to be pulling fluid from the capillary out into the interstitial space. Now, again, this is very negligible, but we must take it into account because it's also acting on the capillary wall. So these are our 4 Starling's forces. 2 of them are opposing each other. One is on the capillary side, and the other is going to be in the interstitial space. So in order to look at the net movement of fluid to see what's really happening in terms of the direction of fluid movement, we have to look at all 4 of these forces, and we have to net them out, okay? Okay, now when we do that, we're going to see that the general net movement that favors filtration is a net movement of fluid from the capillary to the interstitial space. Okay, that's our net filtration. So if we have a larger force that is pushing things out of the capillary, we're going to say that the force favors filtration. And if we have a larger force that's favoring the opposite movement, or movement in the opposite direction, that's going to be called absorption. So again, a net movement of a fluid from the interstitial space to the capillary.